You know when you're at the supermarket and you see those huge strawberries or a watermelon with no seeds? Ever wonder how they get that way? Well, the secret isn't some weird chemical. It's actually a fascinating genetic trick that's been happening in nature for millions of years. And today, we're going to pull back the curtain on the science of polyploidy.
Look, we've got a massive challenge on our hands. Our climate is getting more extreme, and our global population just keeps growing. All of this puts incredible pressure on our food supply. We need crops that aren't just bigger, but tougher, more resilient, and higher yielding than ever. It's a real race against time for plant breeders, and they need every single tool they can get.
So here's our roadmap for this explainer. First, we'll dive into that global challenge. Then, we're going to uncover nature's very own genetic superpower, polyploidy. After that, we'll look at how scientists can actually build what you might call a supercrop, check out the incredible benefits, and finally, peek into the future of farming.
All right, let's start by really digging into the scale of the problem our farmers and scientists are up against. It's about so much more than just growing more food. It's about growing it smarter and a whole lot stronger.
Think about it. Plant breeders are basically fighting a war on all fronts. They need crops that can fight off diseases, that don't care if it gets too hot or if the soil's a bit salty, and on top of all that, they have to produce way more food. It's a hugely complex puzzle, and polyploidy is one of the key pieces helping to solve it.
So what exactly is this amazing tool? It's not some crazy sci-fi invention. Nope, this is a process that nature itself has been using for millions of years. It's a huge reason why we have so many different plant species. In fact, a ton of our most important crops, like wheat, cotton, and potatoes, are natural polyploids.
Okay, let's get a simple definition down. Most animals, including you and me, we're what you call diploid. That means we have two sets of chromosomes in our cells. One from mom, one from dad. Think of chromosomes as the instruction manuals for building a body. Polyploidy is when an organism has more than two complete sets. It's like having 3, 4, or even more copies of the entire library of instruction manuals. And the difference can be pretty dramatic. On the one hand, you've got your standard diploid organism. But on the other, the polyploid, well, it's a whole different ballgame. Having all those extra sets of chromosomes can cause fundamental changes, like bigger cells, which can lead to bigger fruits, bigger leaves, and just generally more robust, hardy plants. So polyploidy happens in the wild, right? But here's the cool part. Scientists figured out how to make it happen on demand. This discovery completely changed the game for plant breeding. It meant they could intentionally give plants these desirable super traits. The secret ingredient is a chemical called colchicine. You can think of it as a stop sign for cell division. Normally, a plant cell gets ready to divide by making a copy of all its chromosomes. But just as it's about to split into two, colchicine steps in and says, nope, not today. The result? You're left with a single cell that now has double the chromosomes it started with. Pretty clever, right?
The seedless watermelon is the perfect example of this in action. Breeders take a regular diploid watermelon plant and treat it with colchicine. Bam! Now they have a tetraploid plant with 4 sets of chromosomes. Then they take that new plant and cross-pollinate it with a normal one. The baby plant that results has 3 sets of chromosomes. It's a triploid, and that odd number of sets makes it sterile, which is exactly why we get that delicious, seedless fruit.
Okay, so we've seen how they do it. But what's the actual payoff? Why go to all this trouble? Well, this is where it gets really exciting, because the results can be absolutely incredible.
Scientists even have a cool name for the most obvious benefit. They call it the gigas effect. Gigas, as in gigantic. All that extra genetic material often leads directly to bigger cells, and bigger cells build bigger plant parts. We're talking larger fruits, more vibrant flowers, and just more plant, period. For any crop where size matters, this is a huge win.
And this isn't just theory. These are real-world success stories. Take sugar beets. Polyploidy means bigger roots, which means more sugar from the same amount of land. For red clover, grown for animal feed, it means more resistance to disease. Even the rye in your bread can get an upgrade, leading to better baking. These are real improvements that have a direct impact on our food.
You know, the benefits are just so diverse. It's not just about getting bigger. It's about creating those seedless fruits that everybody loves. It's about engineering plants that can handle tough conditions like drought, which is absolutely critical for food security. And it can even change a plant's chemistry, making it produce more valuable oils or creating flowers with richer colors.
So, how do scientists know for sure that it worked? They can't just eyeball it, they need proof. So they turn to advanced genetic tests. This graph here is basically a genetic fingerprint. The unique pattern of these peaks tells the scientists exactly how many sets of chromosomes are in their new plant, giving them definitive proof that their experiment was a success.
Now, polyploidy is obviously an incredibly powerful tool, but it's not the end of the story. The path forward means dealing with its challenges head-on and combining this classic technique with the most advanced technology we have.
Right, it's important to be realistic. This isn't a magic bullet. The process doesn't always work perfectly. Sometimes it can make the plant's genetics a bit unstable, and as we saw with the watermelon, it can cause sterility. Now that's great when you want seedless fruit, but it's a major problem if you need seeds to plant next year's crop. These are the kinds of trade-offs that breeders are always juggling.
And this is where the future gets really exciting. The next big leap in plant breeding is all about integration. It's about combining the, let's call it the brute force power of polyploidy with the surgical precision of tools like CRISPR gene editing. Imagine not only doubling a plant's entire genome, but then being able to go in with a tiny pair of genetic scissors and fine-tune specific genes within it. That gives us a level of control we've never had before.
And that leaves us with a pretty mind-blowing question to think about. We're shifting from just triggering polyploidy to actually engineering it. By combining these amazing technologies, could we design the perfect crop from the ground up? A crop with built-in resilience, maximum yield, and packed with nutrition? It makes you wonder, doesn't it? The perfect harvest might be closer than any of us think.